or surgical bone marrow denervation led to the diminished retinal vascular repair in diabetic retinopathy. That's quite a mouthful, but it's a very specific title for a good reason. And I'd also like to acknowledge the other authors that contributed to this paper, Maria, Svetlana, Nat, and my professor, Priyanka. So now before I get into the specifics of my project, I do want to give you a brief overview on what retinopathy actually is. Diabetic retinopathy is one of the most serious complications of diabetes, and it's one of the leading causes for irreversible blindness in the United States for adults. Approximately every year, 24,000 Americans will go blind due to diabetic retinopathy. And after a decade of diabetes, 65% of patients will develop diabetic retinopathy. And unfortunately, current available approaches for the treatment of diabetic retinopathy are really invasive and don't necessarily fully prevent or reverse the pathology. So I wanted to present the pathology of diabetic retinopathy from an ophthalmologist's point of view, looking at these fundus photographs. So on the right here, you can, or I should say your left, you can see a normal view of the retina, so you can see this healthy vasculature. On the left, you can see a retina that has diabetic retinopathy, so you can start to see these leaky blood vessels, some of this hemorrhage that's starting to happen up here. And this next image is of proliferative diabetic retinopathy, so a progressed form of diabetic retinopathy, and you can see that the vascular damage is much, much more extensive in this image than it was in the previous image, with more leaky blood vessels and more hemorrhaging. So a normal person would view these two boys in this image, and I thought it would be interesting for everyone to be able to see that a person with diabetic retinopathy would view this same image this way. So it's characterized by this blurriness, by these spots, and in very severe cases it can cause blindness. And the vascular damage that I talked about in the previous slide is what's causing this blurriness and these spots. So what really is the difference between a healthy individual and a diabetic individual in terms of how they repair damage? Well, in a healthy individual, we have a lot of plasticity. So if we cut ourselves, our body has the mechanisms available to quickly repair that area in a timely manner. But in a diabetic individual, this repair does not really happen, and the mechanisms for this repair are really dysfunctional. So a good question would be to ask in a healthy individual, how are these retinal capillaries repaired? Well, traditionally, when damage does happen to blood vessels, mature endothelial cells proliferate and they repair that damage. And I should mention that endothelial cells is very important, but they are the inner lining of blood vessels, so they're very important to the vasculature. But now there's a growing body of evidence that suggests that endothelial progenitor cells, abbreviated EPCs, actually play a critical role in this vascular repair. So as damage happens, these EPCs, which reside in the bone marrow, go to the area where vascular repair needs to happen, and they differentiate actually into these mature endothelial cells. And at the same time, they induce the mature endothelial cells in the surrounding area to also proliferate and help assist in the damage and repair, or in the repair, I should say. So I talked about how the EPCs reside in the bone marrow. And as the name would imply, the bone marrow resides in the bone. So just to give you a little bit clearer picture so you can visualize this better, but the bone marrow lies in the center of the bone. And as you can see, it's not a simple model. It's very dynamic. It has a lot of dense vasculature, a lot of sympathetic innervation. And these sympathetic innervation, these nerves, are what stimulate the stem cells in the bone marrow to be released to the site of damage and to repair that damage. And the stem cells in specific that we'll be talking about, as I mentioned, are endothelial progenitor cells, abbreviated EPCs. So I talked about bone marrow in the previous schematic, and these are higher magnification images of bone marrow. In the top here, you can see, that this is a control animal, a normal animal, and at the bottom here, you can see a type 2 diabetic animal. In higher magnification, you can see these brown staining tyrosine hydroxylase nerve fibers that surround the blood vessels, and you can see that there's a dramatic decrease in the type 2 diabetic animals. And this graph just shows us that we were able to quantify and measure the amount of TH-positive nerve fibers, and as you can see, there's a statistically significant amount more of these TH-positive nerve fibers in the control than there are in the diabetic animals. And this study was done also in our lab, and they found that along with this decrease in nerve fibers in these type 2 diabetic rats, there was also a decrease in the number of EPCs in circulation, and also a decreased repair also. So that study was really revolutionary because for the first time it showed us that there was some sort of correlation between bone marrow neuropathy and decreased EPC release and then decreased retinal repair. So, but that study really just showed a correlation. So that's really where my job came in, and I was trying to prove a direct causation. So I hypothesized that the bone marrow innervation causes diminished retinovascular repair. And if our hypothesis is true, then the surgical denervation of the bone marrow, which mimics bone marrow neuropathy that happens in a diabetic model, should have the same effect on retinovascular health as in diabetes. So how did I go about that? The methods of how I did this was to mimic bone marrow neuropathy in diabetic animals. In a normal model, by surgical means, we performed denervation surgery on 12-week-old control rats. So the sciatic nerve, which you can see right here, was sectioned close to the exit from the spinal cord on both sides, and then sham surgery was performed on control animals. And we use vascular permeability as a marker for vascular damage. And to assess vascular damage, what we did is after the animals had healed from surgery, we injected them with fluorescent labeled albumin, so fluorescent protein, and we injected them into the rats, and we let them circulate for 2 hours, and we collected blood, and then we perfused the animals to wash out all the excess fluorescent albumin from the blood vessels. And then we collected the retinas. So the areas that would show the most damage would obviously be the areas that were the most permeable and the areas that would show the most fluorescence, and that is what we measured. So I just want to stress here that in diabetes, there's really 2 important factors. There's going to be an increase in endothelial cell damage due to metabolic factors, and then inadequate vascular regeneration due to bone marrow neuropathy. And you need both of these to have retinopathy. If you only have one of these, retinopathy will not happen. So what I wanted to do is I first had a control experiment to test this point. So if I only did the denervation surgery, I really should not be seeing signs of retinopathy because there's no damage that's being induced. And that is what I see here. So in the sham animals and the denervated animals, there was no statistical significance in the difference of permeability in both. So we've now proven that you need both damage and inadequate repair to have retinopathy. So to prove this, what I did was use a technique called ischemic perfusion, and this really mimicked the accelerated damage that happens in diabetic rats. And the technique that we use is ischemic perfusion, which we increase the intraocular pressure in the eye for 90 minutes, normalize the intraocular pressure, and then reperfuse the retinal vasculature. And the beauty of ischemic perfusion is because it only affects the retinal vasculature, we were able to just do it on one eye and then use the other eye as a control. So after we did surgeries on all the rats, we waited until they were healed, and we performed ischemic perfusion, and 2 days later we assessed for permeability. And we did find indeed that bone marrow denervation coupled with ischemic perfusion led to increased vascular damage in the animals that had the denervation surgery versus the animals that had the sham. So in conclusion, we found that direct bone marrow denervation leads to the inability to repair damaged retina vasculature in the retinopathy model. And this is really novel research because now we can use bone marrow neuropathy as a primary target for fixing or potentially having this therapeutic target for diabetic retinopathy. And I have some acknowledgements. My professor, Julia Busick, the graduate assistant underneath it, which I work, Maria Switzlana, our lab manager, Matt, my friends Stefan and Mari, my parents who are here, and the NIH grant for funding this research, and of course the Department of Physiology. So any questions?
Quick question. So these are type 2 diabetes, is it rats?
Yes.
And how did that, how did you develop a type 2 diabetic rat?
We can order rats who have this diabetic model already for us.
Is this the Zucker diabetic rat?
Yes, it is.
So, with your model of like the EPCs, from the damage, is like the mechanism of release versus the mechanism of recruitment of the EPC cells to the cell of the building, are those two separate mechanisms, or I'm wondering like, is it possible that diabetes might affect the recruitment of the cells
to the area damaged.
separately from the innervation and release of the cells into circulation?
I think they're both related in the fact that when diabetes happens, it affects the neuropathy, which then directly affects the release of EPCs. And the release of EPCs is what's happening when you were asking about where there's damage happening, the recruitment of those EPCs. So it's all connected. And I guess diabetes more or less affects the release of EPCs. And so the recruitment of EPCs is more of a secondary factor that happens because of the release that's happening in the bone marrow at standard times.
If you were to deplete cells in the bone marrow with, let's say, cytotoxic compounds, so people who undergo chemotherapy or radiotherapy have injury to the bone marrow, would that have an interaction with preventing healing of a retinopathy?
I don't believe that it would, because it's really the neuropathy, so the nerve damage that's happening to the bone marrow, which is prohibiting the release of these EPCs, or the stem cells in the area.
This is a little off topic, but ischemia reperfusion is often affiliated with oxidative stress. This is more of a general question: is what's known with diabetes versus normal patients with ischemia reperfusion? Are there differences in antioxidant status of diabetics versus non-diabetics, and are there differences in ischemia reperfusion between diabetics?
To be honest, I don't know the answer to that question.
I know it's off topic, but
Yeah.
I was just curious.
I don't know. Julia maybe could answer that a little bit better for you, but I